So thank you, Daniel. So I'm Stanley Nelson. I'm director of the California Center for Rare Diseases. I'm also a co-director of the Center for Duchenne Muscular Dystrophy and work largely in the rare disease space, but particularly with genomic applications towards how we approach rare genetic diseases. And that what I'm going to talk to you about today is advances that we've made and advances that the field is making towards improving diagnostic work for rare diseases using genomic technologies. This is starting to join different genomic technologies to try to make progress a bit more efficiently. And I'll give you a background as we go along. These are my disclosures. I'm an employee of the University of California, Los Angeles, and I'm an ad hoc paid advisor for a number of companies in the rare disease and genomics space. So the overview for what I'll talk about today, and then we can discuss and certainly open to any questions in this regard is, where are we at? How has the field evolved in the last 8-9 years since exome sequencing, sampling the protein coding part of the genome, has become relatively routine and distributed and possible to do in many academic medical environments and in commercial operations. What's the practice look like at the moment? And then where we've been working is integrating transcriptome analysis, RNA sequencing, as best as we can to try to assist the interpretation as we move to whole genome sequencing, identification of rare de novo mutations or rare alleles in the population. And we're particularly looking for DNA variations that are cryptic. That is, it's not clear what the consequence is unless we actually have RNA data to reveal RNA splicing variants or changes to the transcript in rather evident ways, and completely consistent with it being driven by a Mendelian mutation. So a bit different than other RNA splicing talks. And this actually leads very naturally into what are the shortcomings of what we're doing in terms of what tissues express what genes and how do we look and sort through all the noise that we see in RNA sequencing. Excuse me. So exome sequencing was launched at UCLA by Hain Lee and others at UCLA. In 2012, we accepted our first patient into this as a clinical diagnostic. And we've been able to steadily use this in a more maturing approach over the years to diagnose kids with very genetically heterogeneous diseases and solve many of those. In a paper from 2014, after just a few years of this and looking at hundreds of cases, about 800 cases, the diagnostic rate, there are sort of 2 points. And one is that trio sequencing typically unaffected mother and unaffected father, but also affected parent and affected proband or child, substantially increases the diagnostic rate. So we've felt that this was actually core to how we ought to be sequencing, and we've pushed across the field to do trio sequencing as the starting point. And largely that's because it's relevant for determining if rare alleles are indeed compound heterozygous. It's maybe less important for homozygosity, but it can allow greater confidence of determining certainty that a variant was observed, and maybe less important in hemizygous cases. But again, this can also be important in some instances. So it's almost always helpful to have the parental DNAs for a variety of reasons. Predominantly, that's because of de novo mutations, where there's a new disease in the family driven by a de novo mutation causing an autosomal dominant disease. And indeed, the majority of all the cases that we solve in our clinical exome sequencing operation, and this is true for all that have been reported, are driven largely by de novo mutations where neither parent's affected, neither parent has the mutation observed in their blood, but one parent is unknown to be, but is now observed to be a germline mosaic. So that dominates what we do, and that's much easier to determine with trio sequencing. But we have a fairly large number of conclusive diagnoses. It's about 25 to 30% through our operation. There's another portion where maybe one allele was determined, so we might have a potential diagnosis and reduce the search space, and that happens a non-insignificant portion of the time. And then there's some novel gene discovery that's been reported out of this operation over the course of the last years, and that's about 3% of all the discoveries that we make are likely leading to a novel gene discovery, although not all of these have been actually proven at this point. But a majority of the patients, really, we do not come up with a satisfactory answer from exome sequencing. So we're really looking beyond what clinical exome sequencing can do. And we've been looking at this for years now. And part of that was joining the Undiagnosed Disease Network. So we've been part of the Undiagnosed Disease Network since 2014, originally with Eric Verlaine, Christina Palmer, and myself as PIs, and now Julian Martinez has joined and Eric Verlaine has left. And we've been pushing at our site for solution of the genetic cases. How do we do a better job of interpreting the genome? So within this operation and within other projects funded within the California Center for Rare Diseases and the Institute of Precision Health, we've implemented protocols for doing short read whole genome sequencing with Illumina's technology. So, typically 400 base fragments or so sequenced 150 bases from each end, and reassembling that to 30, 40-fold coverage. And either that's generated with partners on campus or partners off campus. And within the UDN, Baylor or Hudson Alpha generated most of the whole genome sequencing. And that has, protocols and analytical approaches to using paired end data, even when the reads are short, as they are with Illumina typically. One can observe relatively small deletions that are difficult to observe by typical clinical microarrays. This is a particular deletion readily observed by paired end sequencing of Illumina's technology, showing many reads indicating a deletion and reads spanning this junction here to this junction here. This particular deletion is clearly inherited from mother and not observed in father, and not observed in others. And it results in a 1 exon deletion of this gene. But other more complex inversions, translocations, gene conversion sorts of events are also possible to observe, and we do observe them within this type of data. We don't tend to find many new single nucleotide variants or indels within the protein coding sequence relative to clinical exome sequencing, but with whole genome sequencing, there is a bit more even coverage of all exons. So there is occasionally an allele dropout from exome sequencing that's remedied by whole genome sequencing, but that's very subtle and has never resulted in a diagnosis, to my knowledge, certainly not in our own work. So this is a perspective of current medical genetics practice, clearly taken from a somewhat more technology-centric approach. And that is, if a medical geneticist can't recognize a syndrome, can't recognize the monogenic trait, and is moving towards panels, increasingly complex panels, microarrays are often performed and certainly have a relatively small diagnostic success rate, but sufficient to be much worthwhile. And often we have cases that have had some single gene and small panel sequencing unsolved for instance, intellectual disability, and hypotonia would be common diagnoses. And microarray is negative. The panels are negative. And in those instances, that's the group of cases that are coming in for exome sequencing. So in our clinical sequencing, exome sequencing operation, we solve about 25 to 30% of these that have already had some prior genetic work. And for the most part, there's parts of the microarray discovery that we would not observe with exome sequencing. And then there's other parts that are overlapped. That is, we can observe some larger deletions, particularly when multiple exons are involved. And then once exome sequencing is performed of the cases that are unsolved over the course of several years, 5, 6, 7 years, with discovery ongoing in the community and with hundreds of new genes being identified every year, we can sometimes go back to look at already existing exome sequencing data and reinterpret it and determine that a single nucleotide variant or an indel or a de novo mutation is indeed now causative, and the report can be addended and returned back to patients and their clinicians. So after reanalysis, there's maybe another 10% that get solved over the course of many years. So it's a couple percent per year at most get solved by reanalysis with ongoing discovery that's occurring in the world. That leaves the majority of all these cases having been through single gene panels, microarray, and clinical exome sequencing, even with the pacing of new gene discovery unsolved. The majority of cases that we work on remain unsolved. And whole genome sequencing holds the promise of observing everything that microarrays observe and doing it better. Finding some of the structural variants that are copy number neutral, for instance, is quite possible to do with whole genome sequencing. So structural variant calling is improved. Single nucleotide indel calling within the exome is also performed, so it does a good job of exome sequencing, and it sequences intergenic regions, 5' 3' UTRs, and intronic sequence as well. But even with all of the discovery, we only find about another 8% of cases solved by using whole genome sequencing with short read technology relative to this typical current realm. And for the most part, whole genome sequencing is not authorized by insurance. So it's not a good mechanism to pay for within our healthcare system. So this leaves us with a bit of a conundrum of how to move forward and how to solve these many, many undiagnosed disease cases where there was sufficient interest in medical genetics clinic and relevance to seek insurance authorization for exome sequencing, microarray, other tests, and still unsolved. So this is a very enriched group to study and to look at. So one of the things that we've learned already is, of course, that whole genome sequencing, because you're looking at intronic sequence and intergenic sequence and promoter sequence, is not easy to interpret unless it's causing an obvious defect to the protein coding sequence. So most of the 8% additional that we solve with whole genome sequence are due to difficult to observe structural variants. But adding in transcriptome analysis or RNA-seq to the whole genome sequencing allows interpretation of the consequence of many of those DNA mutations on cis transcripts, that is on that same allele generally and immediately adjacent to the DNA variant. So this is a rather large sorting operation. And one way to think about it is from a whole genome sequencing perspective. So when we sequence a typical whole genome and sort for minor alleles of less than 1%, there are about 60,000 rare variants. And most of these do not have any obvious effect at disrupting the protein coding sequence. They may be causal for a given disease, but are actually at sort of an a priori level, very unlikely to. Some of them clearly in red are missense, changing protein sequence, loss of function, again, changing protein sequence, essential splice site, canonical splice acceptor donor sequence, also changing the protein coding sequence. So, but that's a very small portion of all that's discovered within whole genome sequencing. And there's an almost equal group of synonymous variants, non-coding, that is in the 3', 5' UTR, and a vast number of intronic variants that are within genes and may be causing a defect. We need other information. So we're going to add in RNA sequencing to augment the DNA sequencing, try to identify all mRNA splice variants that alter protein coding sequence, filter by frequency across the cohort, try to remove common isoforms and noise from within the data, and insist that a given RNA isoform that may be the consequence of a pathogenic DNA mutation is logically caused by a DNA mutation adjacent to that variant. So we're looking at a very cis regulatory manner here. So, of course, all this relies very simply on central dogma. DNA encodes genes. Genes are transcribed into RNA, RNA translated into protein, and we can infer a lot of that. We can't necessarily infer transcription particularly well from the DNA changes necessarily. So we need this bit of data to augment interpretation here and its consequence on the translated protein. So transcriptome sequencing, as I'm sure many of you know, we simply extract RNAs. Most part, we're here looking at poly-A RNA, stranded poly-A RNA, and we're going to sequence the 70 million reads, paired end 150 base sequencing, and realign that back to the transcriptome. I'll just interject that we have a lot of reference information now. So this is GTEx portal, contains 54 different tissues from humans. Some of these are sampled across hundreds of individuals, and they're relatively short, 76 base paired end reads, but very useful for possibly looking at alternative splice forms, but also just what's the abundance in specific tissues, and that can guide us in terms of when is a given tissue going to be of use. But we don't use this to necessarily set what all the isoforms are, and we don't create a reference data set out of this at this point. And instead, we're doing that within the data collected across these clinical cases. So the goal of whole genome sequencing with RNA sequencing is to identify DNA variants and their mRNA disruption. So typically, we would have blood-based genomic DNA purification, and that's performed typically for whole genome sequencing, where we can identify maternal and paternal alleles and de novo mutations and map them back to specific locations, either within exonic sequence or in this pictogram intronic sequence. But this given intronic variant is not necessarily going to be causing a disruption. And so we might expect the canonical splicing to have occurred for this gene. But when we query perhaps blood RNA or a tissue RNA, if that's appropriate, or cultured fibroblasts, making RNA from these, doing transcriptome sequencing as described, mapping those back to the individual exons, we can, in this pictogram, we can see the typical splicing that's expected, and in this particular instance, skipping of this exon, indicating this variant is likely causing, even though it's relatively deep into the intron, skipping of this particular exon. And then you can compare that to all other individuals observed, and indeed, if this is an autosomal dominant trait that's very rare, you can insist that this RNA observation is very rare and insist the DNA change that's causing it is very rare. We can also look at allele-specific expression or just simply aberrant gene expression. This is not dominantly what we're doing. We're dominantly looking at splicing first and then later adding in these others, but really all three are components of this. So I'm going to walk you just through a handful of observations. Many of these are obvious. Transcription, these are different colored exons, and black is indicating the introns. So of course, you splice through, and this would be the typical expected gene product or RNA. And if there's indeed a mutation of splice acceptor, that could result in exon skipping and result in a modified transcript that we could observe. Alternatively, we could have a same splice acceptor loss that has a different consequence, and the splicing could occur within the the reference exon and result in an exon contraction. And again, a similar such mutation could also have resulted in exon extension and making that orange exon larger. So we don't know these things until we look. So even when we deduce that there's a pathogenic mutation, there are many instances where RNA sequencing is actually necessary. A deep intronic mutation can also cause a new pseudoexon to be included into the final mature RNA. And finally, that can also just disrupt the recognition of the intron and result in complete intron retention. So these are a set of the kind of predicted consequences that a given DNA mutation, either a point mutation or a large insertion or deletion, could result in. This is the pipeline that Hein Li, Alden Huang, and Li Kai Wang have worked on and developed in terms of using RNA samples from fibroblast muscle blood, run through fairly typical protocols at this point. We'll talk a bit about some enhanced ways that we're trying to improve this method, generating reads, aligning them back to the genome and transcriptome, and then annotating and quantifying individual protein altering variants by comparison with all of the other samples. There's about 900 individual samples in this now reference database, and even rare patients serve as controls for other rare patients. And then we calculate splice ratios and gather the gene information to try to interpret all of the DNA variants that have occurred. So this would be an example of how this data is looked at. This is the annotated genome with an exon, exon, and exon. And we expect there to be splicing between this exon and this exon indicated here. So these are known junction reads that we expect to see. And then we are searching for novel junction reads that really have never been observed in humans before. And so this is a pseudoexon example where this pseudoexon is now joined to this exon. And then we determine a ratio of the number of reads that we observe here relative to the total number of reads emanating from this. And we do that for every single possible exon join. Alden Huang also tackled a bit about trying to get a bit better outlier analysis for gene expression across data sets that have been generated over a long period of time from males and females from different centers with different growth conditions for fibroblasts, and has regressed out a fair amount of the obvious batch effects to try to observe differences that might be more specifically related to a Mendelian genetic mutation that's causing a substantial shift. And this has proven useful in a few instances. So this is an example of RNA sequencing working in a rather robust way. And this is a 35-year-old with intellectual disability, progressive, but she has a brother, 28 years old, who has a similar such effect, and there's a negative family history. And inconclusive clinical report from whole genome sequencing, no indication of the causal mutation. Sequencing both of these two individuals' RNA from fibroblasts grown in culture from both the siblings, the logic can be pretty simple, right? So in total, there's 500,000-ish total junctions. There's half of these that roughly have sufficient depth to see both, the maternal and the paternal forms. A subset of these are affecting protein coding sequence. A subset are unannotated and harder to look at. And a portion are disrupting splicing in an obvious way. And then a smaller portion are rare. And then when these two are intersected across these two individuals, there's actually only two that are rare and shared. And this overlap of the two in this instance indicated an exon inclusion event that was observed robustly in the proband and her brother, and not observed in a large series of controls. This is all of the other individual junctional reads observed in other samples, and these are the identification of these two individuals. So they're clearly quite unique, and this is indeed the cause of this individual's disease, and could be proven because in this instance there was a biochemical basis for it that could be looked at. I'm going to make a slight side comment on nonsense-mediated decay. So if any transcript has a nonsense mutation in it that's premature, it may trigger nonsense-mediated decay and is somewhat gene specific, but a splice acceptor loss here causing this to be excluded from the mature RNA, and skipping of this blue exon into the yellow exon could result, and actually likely does result in a nonsense mutation if frame shifted in the open reading frame of this skipped message. And this could lead to nonsense-mediated decay of this message. So that creates quite a conundrum for us and a problem for us in that this allele can simply be missing from our analysis. That is, if it was really subjected to strong nonsense-mediated decay, this particular allele with the exon skipping event can be observed at a much lower rate. But if we have heterozygous positions across the RNA at different other positions, we can observe is indeed one allele missing from the total transcriptome and from the relative expression of the two different alleles. So other alleles, they could be in the 5' or 3' UTR or protein coding sequence, can be useful for saying that indeed an allele on the RNA with the premature stop codon is depleted. And this is an example of that indeed led to a rather clear diagnosis of CAPN1, which is an autosomal recessive disease. There was a known pathogenic missense variant in the protein coding sequence that was inherited from mom. And at this allele, 90% of the alleles are observed to be the rare missense variant that is known to be disease-causing. So if it's inherited in a homozygous fashion, it would cause this calpainopathy. And when looking throughout the whole genome sequencing and RNA sequencing, there was a position in the +5 position of this exon that is leading to exon skipping. But the allele with exon skipping, even though this is a very well expressed gene, there's about 1,000 reads spanning every junction, there's only 21 reads, or only about 2% of all the reads are observing the pathogenic variant, making this more challenging to observe unless we could suppress, for instance, nonsense-mediated decay. But in this instance, we can use it because we have an other allele elsewhere in the genome to indicate that this allele with the frame shift is being subjected to nonsense-mediated decay, and this other allele is known pathogenic and clarified the diagnosis in this instance. That's from fibroblasts for calpainopathy. That works because the fibroblasts express CAPN1 actually rather robustly. But that's not true everywhere. This is a couple of examples for Duchenne muscular dystrophy, DMD, one of the most common muscular dystrophies. And even though these are sort of expression levels, so the more blue it is, the higher expression it is in TPMs up here. And indeed, for DMD, it's seen in many, many different tissues, but it's not necessarily the correct isoform that's appropriate for diagnosing the skeletal muscle disease. So this can get complex. And indeed, for DMD, you really have to look at skeletal muscle, even though you could infer from GTEx that maybe we could look multiple other places. In contrast, SEPSECS, which is actually expressed broadly and causes a progressive cerebellar atrophy as an autosomal recessive disease, is seen broadly throughout the body, much less in blood and actually less in skeletal muscle. So figuring out what tissue is most relevant is sometimes critically important. So for instance, titinopathies, really this gene is only needed in skeletal muscle, so it's not expressed anywhere else. And so a muscle biopsy sampling muscle is essential here. OTC deficiency is really only expressed in liver. So there are genes that are very tissue specific that we would have to go after specific tissues that are possible to do. So this does add a wrinkle to this enterprise, but a solvable wrinkle. In the muscle world, we have the benefit of an easily accessible tissue. So one of the things we worked on within the CDMD was creating an enterprise by which core needle biopsies could be robustly sampled with hundreds of milligrams of muscle tissue of adequate histology through a simple transcutaneous approach of 3 different muscles to allow assessment of dystrophin in the context of clinical trials. So we actually have 3 different clinical trials using this approach now. But this allows us to now use this diagnostically across muscle diseases because we can get sufficient tissue from a very small core needle biopsy. In about 10% of cases with Duchenne or Becker muscular dystrophy require muscle biopsy for a complete diagnosis. This is an example of a small child diagnosed relatively late, about 11 years of age, and he was reported to have a partial exon 8 deletion. And he has, one breakpoint is in the Intron 7, and the other breakpoint is with exon 8, and should result in no dystrophin being made because this is an out-of-frame deletion of exon 8. But in reality, what happens when we look at the RNA of this child, his genetic deletion joins part of Intron 7 to exon 8 and extends exon 8 and adds in about 4 additional amino acids. That's exactly 4 additional amino acids never before seen in any dystrophin, and an intron, the part of intron 7 that's remaining, is indeed spliced out appropriately and into this new pseudoexon. So these are sorts of complexities that are needed to sort of get to a complete example of what is really going on, and then is indeed dystrophin expressed or not. So this young boy actually does make some functional dystrophin through this mechanism, and indeed most of the dystrophin that he makes is partially functional. Another type of example in the Duchenne space is a child with a decade-long diagnosis of Duchenne muscular dystrophy from a muscle biopsy, had no dystrophin or very low dystrophin, patchy dystrophin on his muscle biopsy. He had no mutation in the DMD gene by del-dup analysis or sequence analysis. And so, after about a decade of no resolution and no ability to then track what the mutation is in his family, this de novo mutation we solved through whole genome sequencing. But the DMD gene is so large, it's 2 million bases of intronic sequence, that there's actually 6 high-quality, unique hemizygous variants that is family-specific in deep intronic regions in this one gene. And any one of those 6 could indeed be theoretically causal. In this particular instance, one of the 6 variants was de novo and not observed in mother, and it was in a fairly deep intronic position, never observed in nomad. And RNA sequencing on the muscle identified a pseudoexon inclusion, which is identified here, including this and putting the message out of frame as it's an 88 base insertion, and identifying this point mutation as the causal mutation in this instance, and allowing sort of a ability to determine that the mother is not a carrier, and then other sisters of this child to be tested for carrier status. A case with a bit of a different sort of structure to it is SEPSECS. So this is a 2-year-old girl who we demonstrated has 2 pathogenic mutations and a gene for SEPSECS, which encodes for a component of the synthesis mechanism of selenocysteines, and leads to progressive hypotonia, global developmental delay, and cerebellar atrophy. She had 1 clearly pathogenic mutation inherited just as a frame shift within exon 7 of SEPSECS. No pathogenic mutations identified in mother from whole genome sequencing. 2 of the maternal variants, only 2 variants were detected at all in the gene in SEPSECS. 1 was deep intronic, but no splicing alteration was detected anywhere near this. And a second one was a synonymous variant within actually exon 7 as well. And this did lead to exon skipping. So this is the proband, this is the mother and the father. And this exon 7 is skipped about half of all the reads, and the proband skipped about half of all the reads in mother. And this is from either blood or fibroblast RNA. And it turns out that this exon 7 is a fairly weak exon when we look at other cases. There are other cases which skip exon 7 at some non-zero rate, both in blood and fibroblasts. And so, we think it's a weak changing one base within this codon was sufficient to shift it to always be excluded. So this led to a clear diagnosis. It also leads to an issue of how low can we go. So this is a relatively low expression gene. It's observed, but with very few reads, 6 reads observing the pathogenic allele in the child, 5 in the mother, 13 reads with the reference join in father. It is observed in mother and proband, which gives confidence. It's also observed in fibroblasts and blood, which also gives strong confidence that this Mendelian change, the allele from mom to proband, is indeed causal of this. But this is fairly low and quite difficult to see. And so when we look at GTEx and look for different genes for how they're expressed, SEPSECS actually happens to be expressed fairly broadly, relatively low levels, but fairly broadly, actually worse in whole blood. So these are genes that start to get lost in the noise of this method with just, the noise of PCR within amplifying libraries and the like. So we need to do a better job of digging out those low-level genes to actually improve RNA sequencing to observe the consequence of DNA mutations on RNA. So this is one approach that we're implementing. So we've looked at transdifferentiation. Can we make neuronal cells from fibroblasts? And that works to a certain degree, but it's not sort of reliable enough, able to easily scale up. But can we start to deplete high-level expression genes? So if you take, in this figure over here, clearly a gene with a higher TPM, a higher sort of expression level, has more reads observing each junction and makes it easier to observe junctional reads. And genes with lower TPMs have fewer reads. So what we're looking for is, can we take genes with very low TPM? Actually, 80% of all genes known to cause any human genetic disease are observed in muscle or fibroblasts or globin-depleted blood, but at very low levels. So they're expressed. They may retain the appropriate splicing pattern, but they're not expressed robustly. And even a TPM of 1 is difficult for us to see, given the depth of sequencing that we're performing in these libraries. But if we could deplete very high expression genes, which are not on this set, could we move these low-level genes to be the equivalent coverage of a gene that's at a TPM of 10? Can we convert a TPM 1 gene to be more like the coverage of a TPM 10 gene? And how can we get at that enterprise? This is the goal, largely because anything with a TPM of 10 or greater typically has a sufficient number of reads that with 70 million paired end reads, you can robustly observe effects that are occurring on the paternal allele or the maternal allele. So computationally, one can look at all transcripts that are greater than 50, greater than 30 TPM, 20, 10, et cetera. And if one took and depleted all genes that have a TPM of 30 and could get those genes removed from the sequencing library, there would be 90% of all reads removed. So high expression genes are consuming most of the sequencing space. Removing them would allow us to have a larger portion of reads mapping to these lower expression genes. So can we remove roughly 4,000 genes that are expressed at higher than 30 from a typical fibroblast library, and then observe a 10-fold enrichment of low to medium expressed genes that makes RNA sequencing more powerful and gets us to sample a much larger portion of the genome. And to do that, we're partnering with a company, Jump Code Genomics, that is using a method that's been described and put into the literature called DASH, of essentially using for guide RNAs and Cas9 to ex vivo after library construction, deplete that portion of the library that you're not interested in so that you can enrich for the portions of the library that you are interested in. And there's some evidence that this is working. I don't have data to share with you today, but the general idea is, can we enrich for those low-level expression genes and make this method applicable to a much larger fraction of genes, and about double or triple the portion of the genome that's analyzable in each individual fibroblast. So overall, conclusions from this discussion are that negative exomes, that is exome sequencing, where we're failing to identify the cause of a likely rare genetic disease, is really not the end of the genetic workup. It is the end of what's clinically approved at the moment, but whole genome sequencing integrated with transcriptome sequencing is a powerful way to move forward for these cases. And actually, transcriptome sequencing is more important than the whole genome sequencing, but they're really interlocking components. You really need both to what we're proposing here. This is available and can be done at UCLA. It can be done other places as well for people who are needing to pursue this, but it's typically not covered by insurance at this point. There's other ways to get at this issue of how do you improve the transcriptome sequencing. Essentially, if we could completely normalize RNA libraries and get an equal abundance of expression while preserving all isoforms, that would be ideal. Difficult to get to that enterprise, but transdifferentiation of growing cells, either peripheral blood-based mononuclear cells or fibroblasts, are the easiest to conceive of, but one could also work on other cell types that are possible to obtain from humans. It's also possible to just simply promote expression of silenced genes, possibly by demethylation or histone modifications to allow, for instance, patient-derived cultured skin fibroblasts to induce expression of otherwise low-level genes in culture. And then, as we were just discussing, suppress high-expression genes from the final library. We have also been looking at this issue of suppressing nonsense-mediated decay, which is possible to do with great efficiency, such that the mutant allele is more observed and not subjected to nonsense-mediated decay as the inference for the pathogenicity. The most important part is forming a sufficient group that a larger reference set of RNA sequencing performed by whatever the methods are above is grown for tissue-specific types of library, blood libraries, fibroblast, muscle, et cetera, such that one can identify a rare, strong outlier. And that can be done with hundreds of samples. It really doesn't require necessarily hundreds of thousands of samples, for instance. And I don't think we're going to get at this simply by population data from having a larger number of whole genome sequences. A million whole genome sequences won't get us this inference that we need, and we would need to have RNA sequencing to interpret all these rare variants. And then the final thing is having it so that some of these deep intronic mutations that have been assessed with RNA-seq are indeed reported into ClinVar, so that we bootstrap our way to better interpretations of observing what is and is not resulting in a splice change. And then finally, I'll just comment that, of course, some of the technologies that we're using with short read technologies, may get changed to longer read sequencing, which could improve some of these methods, but will still require, even if you could do long read whole genome sequencing, find all structural variants better, you can't interpret most of those without RNA sequencing. So still this integration is going to be critically needed. I'll just end by showing some acknowledgements and some of the team members that have been involved in a lot of this work. I'm happy to take questions or discuss any aspects of this that's of relevance.
Are there any questions? And that's okay, because I've actually, there's something that was separate from your presentation, which was brilliant. Are there, Warren? Oh, sorry.
Oh, yes. Thank you very much. I appreciate it, sir. May I ask your thoughts on, I think you mentioned that increasing the volume of just whole genome sequencing studies may not improve the accuracy because, as you've mentioned, adding the transcriptome certainly increases. May I ask, my particular emphasis is in epilepsy and developmental disorders, and I was wondering your thoughts on doing a prospective study in which you have more gold standard comparisons. So, for example, intellectual delay, you can have a number of clinically administered tests, or if it's, for example, epilepsy, you can compare that to brain imaging. I was wondering on your thoughts on how more accurate this could be with well planned out prospective studies.
Yeah, I do think that I am sort of more of a genomics first or genetics first person. We certainly have many discoveries that we make from just simply looking at the genome. The genome is tractable. The transcriptome is less tractable, but still tractable, right? It's finite, but it may have infinite perturbations on it, though. But I do think that taking and tackling in a systematic way and demonstrating the value proposition is absolutely critical in our kind of health reimbursement world. So I do think that for the clinicians involved and for the discoverers involved, that doing that in as systematic a way as possible, such that it's quite painful to see patients who've fought insurance battles to get to exome sequencing with very capable providers and with a very good rationale, not be able to move past that when most patients don't get solved at that point. We've solved what we can see. We're not solving what we can't see. And so it's pretty obvious how we can get there. So I think it's going to be whole genome sequencing. There's a lot of information in introns, of course, right? Introns are providing information to splicing machinery that I'm an intron. A lot of these things are very obvious, and we've been living this for decades. We now have the tools that we could systematically solve those. So I would applaud, doing things in a way that's, makes the case that perhaps, the most efficient diagnostic route is not what clinicians do at the moment. They do it because that's what insurance companies kind of force them to do. But I do think we should move towards a realm where we simply prove what's most cost effective for our global enterprise. And those sorts of studies would be very important.
May I kindly follow up with, as you mentioned, you're significantly extending on whole genome sequencing because you're adding the transcriptome. What are your thoughts on your approach versus something like Radiogenomics or Sick Kids from Toronto that are utilizing whole genome sequencing? I believe they mentioned that about 40% of patients in their 200 patient study showed that it was quite viable. What are your thoughts on if you utilized a similar approach in infants or in the pediatric population in the same way that they are doing?
Yeah, I would say that's completely compatible with all the data. We now have this looked at in many different ways. Our kind of the way people come to us is through kind of a sequential approach. And when we try to solve with whole genome sequencing, we're still only solving about 40%. So it's not enriched in kind of the newborn populations or the severely sick, like Sick Kids is doing. But so that discovery rate, that solution rate is kind of right in par with what we should expect. And I think what's missing there is not, they're only able to interpret whole genome sequencing that absolutely disrupts protein coding sequence. So a deep intronic mutation that's not affecting in any obvious way, a synonymous variant, an inversion that's within an intron, for instance, can all be affecting the final RNA, but they're not observing it. So they're leaving more data on the table than they're gaining from moving. So essentially, just looking at exome sequencing and moving to whole genome sequencing gets you less than adding in RNA sequencing, but adding in whole genome and RNA sequencing is additive. That is, they're really complementary technologies that allow a more complete resolution. And then it's going to raise issues about what tissue do you sample, right? We're making the enterprise more complex, right? We're collecting an RNA sample. Labs aren't necessarily great at handling RNAs, right? It's a fragile nucleic acid. It needs to be sampled right away. It needs to be stored appropriately. It needs to be sampled appropriately. We have to decide whether you're doing it from blood, or you're doing it from skin punch fibroblasts, then you're growing them for a little bit, right? There's a lot of complexities just by tossing out RNA-seq that makes that arena a little bit harder. But it's certainly true that if you just did whole genome sequencing, you could get to that answer in just a few days, right? It's very simple. It's blood, compared to parents and be done. But be prepared for the fact that a majority of the cases won't be solved. And at the same time, for a relatively small proportion of money relative to whole genome sequencing, RNA sequencing will resolve more. And you know what the DNA change was because you have the whole genome sequencing in hand. Yeah, so I like what Radies is doing, and I like what SickKids is doing. It's just not, it's the right way to go forward, but it will be inadequate. It's just guaranteed.
Thank you, sir. I appreciate it.
Yeah.
Well, we're actually almost out of time. That was really brilliant. I do want to ask something that's a little more general. I was hoping maybe you might talk to us a little bit about the California Center for Rare Diseases. Just maybe some of your goals, some of your needs, the people who are there, and how people can either help, contribute, join. That would be really great.
So the California Center for Rare Disease, we started a couple of years ago, and it's to broadly support rare disease researchers, clinicians, and families who are facing challenges of rare diseases. It's particularly rare genetic diseases. So a lot of the current issues are only a few % of all rare genetic diseases have any therapeutic. So we have to change how we go about doing clinical trials. We want to encourage clinical trials. We want to encourage more powerful clinical trials. So for instance, the core needle biopsy that I touched on a bit is something that then makes doing muscular dystrophy sorts of clinical trials where you want to repeatedly sample restoration of the genetically missing protein possible to do in a more sensitive, appropriate way, particularly since most of these diseases are starting in childhood. We're going to repeatedly muscle biopsy. We need to move away from open biopsies and move to needle biopsies and handle things in a way. So we're looking for kind of systematic solutions that facilitate clinical trial design, facilitates discovery. Any researcher on campus is very welcome to reach out to us and join us. We do have partners that are at other institutions, and we do serve the purpose of trying to solve undiagnosed cases. An enterprise that we're working on is an international clinic that can facilitate whole genome RNA sequencing, and a clinic on campus that's going to mimic and build upon the Undiagnosed Disease Network clinic, where you can bring multiple specialists to have a look at a very complex, multi-system affected patient and possibly reinterpret genomic sequence, but really probably to push what advanced genomics are needed to solve that person and get to a clinical diagnostic, given that we have some new tools for those. And then
what are the
we sample people, we create fibroblasts, we create PBMCs, we consent almost everybody who's in our clinics so that those cell resources and the patients themselves are then resources for making progress in rare genetic disease. So we're really knitting together the clinical world and our basic science world and our genomics world to try to make progress a bit more efficiently.
And what are the criteria you're looking for in partners?
So on the research side, interested researchers that want to jump in and pursue looking at gene discovery. There's a lot of gene discovery that's ongoing where we have clinical phenotypes, but we don't necessarily have the bandwidth to perform basic science work on those cells or on the molecular pathways involved. So we really need experts in those different pathways to step into the rare disease space. And I think, what's very exciting in the rare disease space is there is an enormous amount of activity with therapeutic development. So actually perturbing these pathways in patients is quite possible. Gene therapy, gene replacement is now here. There's, first FDA approvals for gene replacement with AAV is now approved, and others are on the way. There's hundreds of clinical trials ongoing at the moment. So it's a very vibrant space where, basic science researchers can be involved in enterprises. They might not take it to clinic per se. We don't necessarily want everybody to feel like they need to be doing that, but they can be involved in very important basic science research that leads to how do we think about these diseases and how should we be treating them for a really very large group of patients with a wildly heterogeneous set of genetic diseases. And
what about commercial? I was going to ask, what about commercial partners? What are you looking for in those?
Yeah, so commercial partners are very welcome to reach out. We often facilitate clinical trial participation, design. We've helped design various clinical trials in Duchenne and other muscle diseases in particular, but that's true across a number of different rare disease areas. So we can knit commercial companies with expert physicians or researchers, depending on what's appropriate. And I think in other places, and we work with other programs that are developed on campus. Like I think a couple of your researchers were presenting on projects and components of the Center for Duchenne Muscular Dystrophy, which is itself a rare disease, but it's a relatively common rare disease. So there's lots of clinical trial activity ongoing there, and there's lots of need for a serious, ultimately fatal genetic disease. So we try to just simply build teams that can make progress better, that involve UCLA. I think that's probably the common denominator.
Sure. It seems like Los Angeles is definitely a center for this kind of research.
Yeah, I think we're trying to make it more and more of a center. I think there's a huge need, and I think we can, one of the benefits of such a large medical enterprise as UCLA and UCLA Health that's integrated with academic, these families don't necessarily have a good place to turn to for diagnosis. It does not occur out in the community very robustly. So it really needs a very academic institution. And then as those diseases are solved, you don't want to just drop it. You want to sort of hand it off to researchers, which UCLA has. So I think we're in a very good place. And with partner institutions in LA as well, Caltech, USC in particular, on the research side, there's really a vibrant world for moving some of these ideas forward.